Hi kids, Dr. Josh here. Today we're going to learn about DNA. The reason I know so much about DNA is because one day I was feeling lonely all by myself, so I decided to clone myself. I call it Pamela. Wave to the cameras, Pamela. She's only a few days old. Well, today we're going to learn about this, DNA, and how it's replicated, transcribed, and translated. Replication starts out with a DNA molecule. Each strand is made up of nucleotides, which each contain a phosphate group, a nitrogenous base, and a 5-carbon sugar, called deoxyribose. These bases will pair up with their complementary base pair using hydrogen bonds, adenine with thymine, and cytosine with guanine. To replicate the DNA, a helicase will separate the bases into 2 strands, like a zipper. Then, single nucleotides will come find their complementary base pair and be attached by a DNA polymerase, gradually forming another DNA molecule. This replication is called semi-conservative because half of the DNA is always being conserved. The conserved strand is called the parent strand, and this one is called the new strand. Transcription starts the same way, with a helicase separating the DNA strands. However, this is only done to a section of the DNA molecule where the needed information to create a protein is located. The single nucleotides begin pairing on only one of the DNA strands, with the help of an RNA polymerase. When the pairing is complete, an mRNA strand is formed. In this strand, thymine is replaced with uracil, and the 5-carbon sugar is ribose rather than deoxyribose. The strand the mRNA paired with is called the antisense strand, while the other one is called the sense strand. Translation occurs inside of a ribosome. It begins when an mRNA becomes attached. When it does, transfer RNA begin coming to match their 3 anticodons with a 3-codon group of the mRNA. Each transfer RNA has an amino acid attached to its end, and if the anticodons match the codons of the mRNA, it will leave behind the amino acids, creating a chain which is called a polypeptide. These amino acids are attached by a peptide bond with a catalyzed condensation reaction that is done by an enzyme. When the mRNA has been completely matched, the polypeptide is complete. She's still learning to talk.